Good morning, everyone. Welcome back. Thanks for joining us. We're glad you're here. I'm Steve.
And I'm Scott.
Together, we're Backyard Musings, Science and Technology channel.
Yep, we hope you had a good weekend, and we are talking about a science, a health issue. So let's get into it.
Yeah, this is a strange one. Scientists at Fred Hutch Cancer Center have reported an important advance in the effort to neutralize Epstein Barr virus, EBV, a virus believed to infect about 95% of people worldwide. EBV has been linked to several cancers, neurodegenerative disorders, and other long-term health conditions. Sounds nasty.
I know somebody that has this.
Oh, wow. To tackle the virus, researchers used mice engineered with human antibody genes, not quite humanized mice. This approach allowed them to generate fully human monoclonal antibodies designed to block two critical viral surface proteins. These proteins normally help EBV attach to and enter human immune cells. The findings published in Cell Reports Medicine on February 17, 2026, highlight one antibody in particular that protected mice with human immune systems from infection when they are exposed to EBV. Would you like to know more?
Yeah, this person that has this, she's an older lady. I also know somebody that is probably in their 40s to 50s, and they were diagnosed with this a few years ago. And it's almost like mononucleosis type thing.
Oh,
But I think it's worse.
constantly tired?
Yeah. Okay, quote here. Finding human antibodies that block Epstein Barr virus from infecting our immune cells has been particularly challenging because, unlike other viruses, EBV finds a way to bind to nearly every one of our B cells. B as in boy. That's explained by Andrew McGuire, PhD, a biochemist and cellular biologist in the Vaccine and Infectious Disease Division at Fred Hutch. We just, again, going on with the quote, we decided to use new technologies to try to fill this knowledge gap, and we ended up taking a critical step toward blocking one of the world's most common viruses.
One of the central obstacles in this research was identifying human monoclonal antibodies that could stop EBV without provoking an anti-drug immune response. Such reactions are common when patients receive antibodies originally produced in other animals.
So they use those monoclonal antibodies during COVID, I think, right? Wasn't that one of the medicines that they tried to use to fight off the COVID virus? So
I thought it was ultraviolet light that they injected in you. That's not. They didn't do that.
I don't remember. Yeah, I don't remember. Maybe it was drinking bleach. I don't know. The team concentrated on two viral proteins. The first, GP350, enables EBV to latch onto receptors on human cells. The second, GP42, plays a key role in allowing the virus to fuse with and enter those cells. By using a specialized mouse model containing human antibody genes, the scientists generated two monoclonal antibodies targeting GP350 and 8 targeting GP42.
Additional studies conducted with support from Fred Hutch's Antibody Tech Core identified specific weak points on the virus that may guide future vaccine design. In laboratory tests, one antibody directed at GP42 completely prevented EBV infection in mice with human immune systems. An antibody against GP350 offered partial protection.
Each year, this is staggering, more than 128,000 people in the U.S. receive solid organ or bone marrow transplants. These patients must take medications that suppress their immune system to prevent rejection. Currently, there are no targeted treatments that reliably prevent EBV infection or reactivation during this vulnerable period.
I wonder if they found this out, that it rejects, that you need medicine, when Dr. Frankenstein put his monster together. And you'd think those amputations, and
he had parts from everybody, right? He had parts from
he had parts from everybody. So he had to get over that hump of rejection,
Yeah.
Huh. Uncontrolled EBV infection after transplantation can lead to post-transplant lymphoproliferative disorders, PTLD, and aggressive and sometimes life-threatening lymphoma. PTLD most often develops when EBV spreads unchecked in patients whose immune defenses are weakened.
Transplant recipients can acquire EBV if donor organs or cells carry dormant virus. Patients who were previously infected may also experience viral reactivation when immunosuppressive drugs weaken immune control. Children undergoing transplants may face especially high risk because many have not yet been exposed to EBV and therefore lack natural immunity. That's a big deal, right? That's a big deal nowadays. I've heard that natural immunity is going away. People are losing that ability to fight off some of these diseases, and that's why there's a rash of stuff. And yeah, we just don't have the immune systems that we used to.
I think it's bovine injections. We drink the milk.
Oh, hey.
It wasn't that funny.
Don't be knocking on the milk.
Right? You like milk. That's right.
I do. I just drank a glass of milk before we went on. So
the researchers are working toward a therapy that will involve infusing these monoclonal antibodies into high-risk patients. If I was at high risk, I'd try me. The goal is to block, as long as you give me $50 a month, I'd be fine. The goal is to block EBV infection or reactivation and reduce the likelihood of PTLD. Fred Hutch has filed for intellectual property protection covering the antibodies identified in the study. McGuire and Chan are collaborating with academic partners and an industry collaborator to move the potential therapy forward. If development continues successfully, the treatment will first undergo safety testing in healthy adult volunteers before advancing to clinical trials to transplant recipients and other immunocompromised patients.
Bring on the clinical trials.
Let's try it.
Yeah.
See if it works.
I'm sure that there's got to be people out there who would just say, look, I will volunteer and, just take my chances. What do they got to lose? So anyway,
isn't that Trump's line? Trump said that. What have you got to lose? Vote for me.
I got to lose. Other than the side effects, and we all know about the side effects, and we don't need to get into that. So, But anyway,
right, folks, thanks for joining us. We'll catch you in the afternoon.
See you this afternoon. Thank you.